Hi, everyone. Okay, so it's a tremendous honor, but frankly, it's also an impossible task to introduce Richard Durbin, our second keynote speaker. For all the issues around citation counts, when someone has 300,000 of them, you do start wondering sort of how long of an introduction you're going to have to compose to begin to cover their contributions. To quote his certificate of election to the Royal Society, he played a leading role in establishing the new field of bioinformatics, enabling genomics to prosper. So given such immense impact, it seems a bit unnecessary for me to recapitulate his biography and list his many awards. I will say that he's apparently very loyal to Cambridge, England. He completed most of his studies there, and he continued to work there and at the nearby Sanger Institute ever since. His loyalty extends to this meeting, which he attended before it even had a name, coming, I think, close to yearly to ask thoughtful questions and engage in long discussions at the bar, helping to establish it as a nexus for probabilistic methods in genomics. Thankfully for all of us, he's a sort of less steadfast when it comes to scientific topics. And so he's switched between fields of biology entirely every decade or so, making central contributions to each of those. Many of you will be familiar with his work on databases for genomic data, PFAM, methods for genome assembly, SNP calling, imputation, just to list a few. What you may not know, and I was reminded of just 2 minutes ago, is that he did a postdoc in the psychology department at Stanford, or that his PhD was actually focused on analyzing nervous system development and connectivity in C. elegans. So it was never published, this PhD, but it so happens that my neighbor at Columbia, the neuroscientist Oliver Hobart, keeps a copy of it. So I asked him about it by email, having told him that I was about to introduce Richard, and he replied, his PhD thesis is a total legend in the C. elegans world. It's required reading for anyone who does developmental neuroscience and or circuitry in the worm. Everyone is totally baffled by why it was never published, which makes it the most read PhD thesis in the worm field. If I were to introduce him, I would put him on the spot and ask him, why did you never publish this, you lazy bum? This is where you have to imagine Oliver Hobart saying that, not me, by the way. Richard's most recent foray, luckily for me, is into population genetics, and as his point of entry into the field, he revolutionized the study of demographic inference in humans and other species with the development of PSMC and MSMC. In fact, I myself first met Richard, I think 13 or 14 years ago, when we got stuck together on a suspended ski lift during a free afternoon at a Keystone meeting. And with our sort of skis dangling in the air, he came to realize that I was a population geneticist and shared with me the basis for what became PSMC. Now, I have a nagging suspicion that being a bystander at that moment will end up as the highlight of my career. His most recent interest is in the oldest and most central problem in evolutionary biology, speciation. And once again, he's transforming how we think about this process through his study of adaptive radiation in cichlids and also in connecting what we've inferred in humans to metapopulation dynamics across the tree of life. So I'm going to stop now and let him start. But before I do, in preparing my remarks, I noted this habit of changing fields once a decade, and also that it's been a decade since he entered population genetics. So if you have any unsolved, super interesting problems you care to throw his way, please do. Okay.
Okay, well, it's very unfair. Yeah, so I have a sort of, I'm poised between familiarity and fear, basically. Familiarity for the reasons you say. Actually, came to my first ever meeting at Cold Spring Harbor in 1983, so it's 40 years ago, and come more or less annually ever since, I guess. And I know the people, many of the people at this meeting and this meeting, but fear because there's all these incredibly smart people and I feel like I'm getting behind. I need to switch fields. So, and so many of you in the audience know more about the things I'm going to talk about than me, I think. Anyway, I'm going to talk about PSMC. Sorry, I changed the title a bit. There's a bit of fish and speciation at the end. I'll try and transition into that. But I want to start by actually what I'm spending a lot of my time on now, which is, I think in a way when I look at my career, at least over the last 30 years or so, it's been involved in sequencing genomes, and people tend to think that that's done, and actually a lot of the great things at this meeting are all about all the things we can do with building on genomic data. But I think, I think you'd all agree it's a transformative technology for biology. But I think we're still, we underestimate it. We're still in the middle of gaining fundamental genomic knowledge on which we will be able to build even better things in the future. So, and I see it as phases, and the phases are basically driven by technologies. So the first phase was sequencing the human genome and other key organisms, and that was with Sanger sequencing, which actually, almost is dead now. They're not quite, but is what started DNA sequencing and was automated. And as we all know, cost billions of dollars for single genomes. And then in phase 2, there was Illumina short read technology, which is in a way the predominant technology now. And that allowed us to do population resequencing and also genomic assay by sequence, all these star-seqs and so on. And but that gives short reads. And now, as you all know, we have single molecule long reads. Actually, we haven't heard very much about data of that sort. But the costs, just as 10 years ago, the costs of Illumina data were dropping fourfold a year. The cost of PacBio data, the accuracy is improving all the time, and the cost is dropping fivefold this year. And I think, that is going to be transformative in the future. And in particular, it really lets us get directly at molecules. They aren't copied in any way. And we can get 100-fold longer reads, and that enables us to sequence, assemble new genomes. And I think we're still at the start of that phase, and it's going to change the next decade in the same way that in each of the last decades have been sort of really changed. In particular, this ability to assemble new genomes allows us to go and sequence almost anything, and that cost is dropping rapidly towards $1,000 for an arbitrary genome. It was a big, and Harris Lewin and others, and group of people I got connected with. In some ways, the most inspirational meeting I ever went to actually was a meeting of people who were sort of just drooling over the possibilities of getting genomic data on their organisms, and they knew all about all these amazing creatures which I didn't know anything about. And it seems daunting. There's actually about 1.5 million, 1.7 million, I think, named eukaryotic species. That seems daunting, but it's not. We've sequenced several thousand at twofold improvement a year, which is conservative for sequencing technology. 2 to the 10 is 1,000, so we can scale 1,000-fold over a decade, and we can sequence over 1 million species. The trouble will be to find them, but we can essentially get complete coverage of the living world, and that will enable us to do all sorts of amazing things, and it'll enables us to work in genomic ecology and conservation, of course evolutionary studies, engineering, all sorts of things. So this is a sort of lengthy introduction. I'm involved in the project in Britain. I think it's logically going to get done by region, so we don't all fight over doing the most important things. And to sequence all 60,000 species in Britain and Ireland. The idea is to start, we're very collaborative, part of the Vertebrate Genomes Project with people in Rockefeller and people across the world. But it simplifies logistics for us to work in the UK. There's a huge amount of knowledge there. We can get partners to collect things. And we're having to work with museums and botanical gardens and things that are regarded as slightly fusty, I think, by a lot of people. And I've got a new appreciation, actually, of what they do. And I think rediscovering the diversity of life is an amazing thing. So there's a big consortium. Sanger Institute is involved in the sequencing. Mark Blackster leads it. And this has really been scaling up. By February, we already submitted 700, but we've got more data on 300, so there's 1,000 species for which we've already got complete data sets. We've got another 2 and a bit thousand in progress with some sequencing data. So it's overall 3,000 from 864 families. And this is all the way around here. You can see all of these are underway. All the red things are sequenced. I don't know what the lengths of the bars are. I can't remember. But they go from plants, fungi, metazoa, deuterostomes. These are arthropods, lots of insects. There's a lot of insects in the world. Okay. And here we sequenced mistletoe, sort of as a demonstration project. That's got a 99.5 gigabase genome. It's a bit disappointing. We didn't break the 100 barrier, but that's what's there. It's got a 10 gigabase chromosome 1. This is the automated assembly and scaffolding just with PacBio data and Hi-C. There are some problems in this, and there's some stuff here which we call shrapnel, and there's some small bits. This is after a little bit of patching up. And it's got the right number of chromosomes, and there's all sorts of interesting structure about the way the chromosomes are organized. So this is essentially a Hi-C map, so lots of stuff in there, and a lot of transposons. Okay, so lots of new genome sequences. So this is what can we learn from single genome sequences? They're actually not single. We're getting data from a single organism, but most organisms are diploid, so we get variation data from a single organism. And modern assemblers assemble both haplotypes in a diploid. Or if they don't do that, you at least get one really good contiguous chromosomal reference, which until the last couple of years, these are mostly better than the human reference was, until the T to T was completed. So we do have variation information. We have a sample of 2 across the full genome. And it's surprisingly informative, and I'm going to talk about this PSMC method that many of you know about. But I have other ideas, and I think it's good to think about what else we can do. And I think there are other population and genetic things that we can do with single diploid sequences. And it's a good drinks topic because there's going to be a lot of data. Okay, so how are 2 sequences from a species related? In the 40s, Gustav Maleko introduced this concept of coalescence, of thinking that there are 2 separate sequences. This is at one point along the sequence, and they're different, but they each have a parent, and those are probably different. But at some point, there'll be a common ancestor from which they derived. And we can think, going backwards in time, of coalescence, of these 2 lineages coming together and forming one, and there's a time to the most recent common ancestor, which we'll call T. And if we have a population with size n, so there's n possible parents, you can think of each of these copies, in some sense, choosing a parent at random from the population. So there's a 1 over n chance that they'll pick the same parent. So that's the standard model. And that immediately leads us to this picture that the TMRCA is exponentially distributed under that model with mean n. And it's a pretty good model. There are assumptions involved. And so if you've got a mutation rate mu, and the time is t in generations, and mu is mutation rate of mu per generation, then the probability of there being a mutation at this position, or the expected number of mutations, will be 2t mu. And given that mu is small, 2t mu is almost certain, is going to be also pretty much the probability. So that's at one place, but if you look along the genome, coalescence times vary. So here we're running along. This is simulated data from MS Prime, because we don't know the truth for any real copy, but this is under the model. And it's piecewise constant, so there'll be pieces and then jumps to another time and so on. So you can see it varying here. Quite variable, okay. And so we have segments of fixed TMRCA, and they're separated. And the reason why they're separated is because there have been recombination events in a parent, so that the sequence in some individual to the left of the recombination point came from one parent, and or in an individual came from the chromosome inherited from one parent, and to the right in the gamete that they're going to pass on came from the chromosome inherited from the other parent. And so these are 2 different individuals, the 2 parents, and they have their own histories. And they have a common ancestor, and so we're switching and going from red to blue at this point between one history and a different history. So that's what gives rise to these. And we can also look at this picture and we notice that there are some regions where the TMRC is recent, and of course the density of mutations is low there because there's not much time to accumulate them. And then if the TMRC is a long time ago, then there'll be more time, and so a higher density of mutations, and you'll get a higher density of mutations there. And so actually, if you look along the genome, mutations don't appear at random. They're clustered. And that clustering gives us information. And it gives us information about the age of the segments in that region. And the age of the segments tells us something about the population, about what was going on in the population. And that's the key to PSMC. So PSMC is essentially just looking at the differences, because that's all we see when we see a pair of sequences. We can compare them and see the differences. And it's using that to make inferences about the possible time of the most recent common ancestors in the past. And we can think of that as hidden information. There's a state. There was a most recent common ancestor. We're not sure when it is. And so we've got a sequence along here, and we've got a hidden state, and that corresponds exactly to what we need for a hidden Markov model. So we can actually consider all possible hidden times, hidden states, and these are transitions, jumps from one state to another state. So Hung Lee, when he was a postdoc with me, before he went off and did amazing things in providing the foundation for all sorts of genome bioinformatics software, developed this into a working model, and we can use all the standard machinery of hidden Markov models, Baum-Welch and so on, to fit the parameters. And what are the parameters? Well, we know about the emissions. The emissions are the things you observe, and that's this 2 mu t, where t is the time. And then there's the probability of recombination, and that's 2 rho t. So the probability actually of a jump, of one of these jumps happening, also depends upon t, because at some point between here and here, there will have been a recombination event. And so that probability depends on t in a straightforward way, but where you jump to doesn't. But luckily, the whole standard coalescent theory explains where you jump to. And this looks slightly scary, but it's not really scary. So we can, from standard theory, we can say what the coalescent, what the distribution is of places that you will move to. It's this Q transition matrix, Q of the new coalescent time t given the old one. And there's one sort of set of parameters, these lambdas, and the lambdas are essentially the population size. We had this n. So we've got a system which is determined by the set of population sizes in the past under the model. So we can fit that. So we run this model. Here's simulated data, the black line. These are the posterior distributions. You can see it fits quite well. We model the time, although it's continuous, or, well, I suppose there are discrete generations, but we can't afford to have 200,000 of them, so we have bins of time. But all this works. And if we run it, looking now back in time on the x-axis in generations, and looking in blue, we see this is the number of coalescent events that happen. And what we see is there are times when there are a lot of coalescent events, and there are times when there are relatively fewer. And if there are a lot of coalescent events, the chance of a coalescence event is inversely proportional to the size of the population. So we can infer the population size at that time as 1 over the blue, basically. It's the inverse of the rate of coalescence. So we infer the effective population size as a function of time, and that's the red line, and the green is a bootstrap where we block bootstrap. So that's the basic PSMC model, which many of you know. And you can see it's kind of not very well defined at this end or at this end, because most of the coalescence events happen in this period, and there aren't very many at the ends. Okay, so we can apply that, and this is not actually in the first paper, but fairly soon after, to a range of different humans. And what we see is a a dip here. Actually, if I go back, this was one individual. We had a kind of dip here. We thought there was a relatively low population. That's a bottleneck, a population bottleneck, and that corresponds roughly to the out of Africa, well, we believe to the out of Africa bottleneck, because populations that came out of Africa all share their history behind that, to the right. They're different here, and they all go through this bottleneck. And then these ones here, which don't have such a deep bottleneck, are all African populations. So there's lots of things that are nice about this. We can also take ancient samples, where we have a diploid sequence, which we do for both a Neanderthal and a Denisovan, and Hung did this when those appeared. And they separated about 500,000 years ago from what became modern humans. And they appear to have had a small population size for a long time, and that's actually consistent with them having low diversity and a whole set of other properties. So that's kind of interesting. So there's a set of things we can see there. Okay, so what's happened? So since then, in the last decade or so, there have been a lot of advances in various ways. One is a kind of cleaning up of the model. We need, and I'm going to come back to this actually later, we need to consider recombination events where there's a recombination, but the common ancestor turns out to be the same as the one it was to the left, so that you don't change the common ancestor. And that's a correction that's called the SMC prime model, and it fixes some non-convergence or some things we had to fix in a slightly hacky way in the original model. Then there's a very nice observation by Kelly Harris when she was with Rasmus Nielsen and Yun Song, that normally the number of, in a hidden Markov model or in a Markov model, you have a quadratic number of transitions possible. There are n states, or we'll call it T states, capital T. You can go from any state to any other state. That's actually true in this case. You can transition from any time to any other time. But we said that although there's a quadratic number of these, it's parameterized by T and S, there's only actually underlying it one set of parameters. And the way that it happens is that you get a recombination event at some point. And so all the different times here could recombine down. We can basically condition on the recombination time. And the recombination time is a linear thing. And so you can in fact calculate the update in linear time rather than quadratic time. And that there's an algebraic exact formulation for that, and that speeds things up by an order of a factor of however many values of T you have. And then we started exploring multiple samples, and there's a paper here, Stefan, it's very quite complex, but it required more approximations. So he looked at the first coalescence time, and there was a set of analyses comparing different populations, and it fit the first coalescence time. But actually, it turns out that it's better to use a composite likelihood to just take lots of pairs and treat all the pairs as independent. And there are various other things that are done this way, like a lot of the way that we look for recombination hotspots. And that turns out to behave better, and there's a whole machinery and set of things which I'm not going to talk more about here, but built on that, which Stefan in particular has built. So I want to give you two stories about new things, because everything so far a good fraction of the audience knows. So the first is from Regev Schweiger. This is in bioRxiv. It's accepted at Recomb. And this is a sort of ultra-fast version of PSMC. And it's always good to have things that are fast. In fact, one of the things I say that's a driver for bioinformatics, since sequencing data quantities increase twofold a year, and Moore's law is twofold every 2 years, we have to get faster and smarter algorithmically every year, basically, in bioinformatics. And luckily, because the more data we have, the more it looks like things we've seen before, there is scope to do that, but it's a key driving principle. So So PSMC is a hidden Markov model, and for those, you basically have hidden state, and these are your observations. And we're moving from left, and the way that works, the algorithms work, is the dynamic programming algorithm. You can run from left to right, and you can obtain a distribution. Well, it's not a full probability distribution. It's a function over the hidden state values, the coalescence time of possible values of what you think, conditional on all the data seen to the left, is the state, essentially. It's the information about the state from all the observations to the left. And then you can do the same from the right, and you can run from the right, and that's called the backward algorithm. And essentially, it's a mirror symmetric situation, and you get whatever your knowledge is about this hidden state from all the observations to the right. And then you can combine those together to get a posterior for the value at the hidden state by essentially multiplying those two. So that's the fundamental way of operating. And all those operations in general have to be over the state. And so, as I said before, we sort of discretize this typically into something like 50, 32, 50, 64, 100 values. And of course, you have to maintain all the memory and carry out calculations for each of those bins. Okay, so PSMC and MSMC squared were order of the length times t squared. Then there was a sort of improvement where you just looked at the het sites and you could jump. You didn't have to go block by block. And then Kelly Harris introduced dical, and this dropped the quadratic dependence on t. And Pierre Palomara sort of combined these two things and did some other strong coding. And so I think this is the state of the art in terms of speed for the implementation up until now. So what did Regev do? Well, he observed that if you look at these posteriors, if you look at these distributions, they tend to look like gamma distributions. And for reasons, so that's empirically an empirical observation. They're very well fit by gamma distributions, almost all of them. That you actually do get little bimodal ones, but we'll ignore that. But you can quiz me about it. So, and the gamma distribution is a very nice distribution. Just has two parameters. And so Regev's idea was just to store those two parameters, to represent your information that you have as in the form of a gamma distribution, just with the two parameters alpha and beta. They empirically look suitable, but also they're conjugate to Poisson emission probabilities, which is the natural thing. So it's a very natural conjugate prior in some sense. And you can get a pre-calculated update rules for both the transitions and the emissions. Well, the emissions actually are very, very natural. They just increment the alpha and beta parameters in a natural way. The transitions we deal with by doing a one-off calculation based on the current model and we linearly interpolate on it. And so we have an interpolation operation in a 2D space. But that's a fast operation, but we can make it very fast. So then you can have an update rule which just runs along updating these two parameters, and it's independent of the number of time points. So it's significantly faster. So this is compared to MSMC2. This is a logarithmic scale. This is per genome. So hundreds of seconds for MSMC2. This is the number of time intervals. Of course, it's flat for gamma SMC. So it's kind of... SMC is fast, but we're at least tenfold faster, and in fact sub a second per genome per pair of comparisons, and that allows us to compare hundreds of thousands or millions of pairs of sequences. If we look at the accuracy, this is the true TMRCA according to the model, and the estimated one. There's ASMC, MSMC, and gamma SMC. they all are pretty good. This is slightly better, slightly higher R squared, actually higher than MSMC2. I'm not sure whether it's using, ASMC allows you to use additional information. I'm not sure quite where that's coming from. But they're all mirroring the same things. If you notice, some of the places where this MSMC2, which is kind of a classic PSMC implementation, is higher, our version gamma is a bit more conservative, and also perhaps will be a bit more conservative on the low side. And this is something that Trevor referred to on the first day as a bias. So he referred to it as a downwards bias here compared to the truth, but it's a bias in the posterior mean of the estimate. And actually, it's not really a property of PSMC. It isn't saying, I don't think it's correct to say that PSMC is biased. I think it's a natural consequence of Bayesian estimation. So in Bayesian estimation, which is essentially what we're doing, we're using all this information to update our posterior estimate of what the coalescence time is at each position compared to our prior, which is an exponential, or for constant n. Bayesian estimation, you're combining prior and data, and you're always biased towards the prior in the mean. And I think since actually we hadn't thought about this, I think it's useful to think through this example. So it's a little toy example, which is actually quite a good model for what we're doing. If you think of a Bernoulli random variable x with an unknown probability theta, which has got a prior which is uniform on 0, 1, so you're going to pick a random theta, which is the probability between 0 and 1. That's the model. And we note that the uniform prior between 0 and 1 is also beta of 1, 1. If we sample one observation from that now, what's our posterior estimate or expected value of the posterior mean? Well, if we sample a 1, then our best guess is going to be this. We've got this prior, it's going to be, using pseudo counts, it's going to be 2/3. And if we sample a 0, if our coin flip with our theta, then our best guess for theta afterwards, the posterior mean estimate, is going to be 1/3. So the expected value is going to be theta times 2/3 plus 1 minus theta times 1/3, which is 1 plus theta over 3. And that's biased towards, in exactly the same way that this is. And if you've got more data, it will be less biased. And actually, the same is true here. If we have more mutations, then this is less biased. But the answer to this actually is not that the whole thing is biased. It's that we shouldn't be using the posterior mean until we have to. We should be using the entire distribution. And luckily, and we can do that. We can afford to do that because our forward-backward process actually evaluates over the entire distribution. And when we estimate the parameters, the n's, the population size parameters, we do that over the entire distribution. So this isn't damaging, and in that case, you're not hurt. So, and you shouldn't use the posterior mode either, the most likely thing. Okay, so I'm sorry, I know I'm sort of, you all know that already, but, okay. Okay, so this is, these are just things we can do, and RegEv's only really started to explore this, and we've started to apply it to bigger data sets. This is with 1,000 genomes. You get all sorts of fine-scale population structure. You can also, this is the mean. Of course, you shouldn't be doing that, but have to show something at each point. This is using the distribution. We can look at, how much density there is within the last some amount of years, 5,000 years or so. So you have a distribution there with gamma SMC, just as with a normal PSMC. Okay. And we can look at the distribution of coalescence times along the genome, and we can see regions where there are lots of recent coalescences, and this is around the lactase region, and there's lots of things here. There's all sorts of cool patterns and structure you see. And this is basically Pierre Palamara and colleagues, Alkis Price and others, Jonathan, who I think is here somewhere, did this in 2018. So this is kind of copying that. And we can scan across the genome and find peaks. I'm not sure what's going on here. Maybe somebody can tell me afterwards and do the same as that. Okay, so that's one thing. There's a new tool. You can do even more SMC operations, and that can allow you to do things which you might not have thought of before. So the second is Trevor, who talked on, second thing I wanted to talk about is from Trevor, who talked on Wednesday night. And this story starts, actually, when we first talked about PSMC, I actually can't remember whether it was Hung or me, but I remember that Molly asked a pointed question and pointed out that one could get this sort of bump that we saw in the humans before the Out of Africa event, that you could get an apparent increase in population size, which is a reduction in coalescence rate. This population size is 1 over the coalescence rate, not by actually increasing population size, but instead by splitting the population into separate pieces so that the things that are in the two pieces can't coalesce with each other. Okay, so PSMC measures 1 over the coalescence rate, and the population structure in the past can also change that. So following Molly making that point, the advantages of giving talks with lots of smart people, we did some simulations where we had population structure, and we showed that, by splitting into 3 populations all of the same size so that the total count was the same, we could actually get a bump that looked something like the bump that we see. And that's from the supplement of the original paper. But then, later, Olivier Mazet and Chiki published a couple of papers showing that actually, by manipulating the subpopulation structure and the migration rates, you could keep a constant total population. I think you can keep each of them the same size, actually, n. That's what all the n's are here. And end up with any curve that you want to. So that's a sort of extreme case showing that there's a sort of exchangeability of structure and measured coalescence rate. So that was an n-island model. So you can take that 2 ways. You can either say, oh, well, this means that the whole stuff is meaningless, or you can say that, well, you're still measuring something. It is 1 over the coalescence rate. There is a coalescence rate, at least it's, under this model, and that's what NE is. In fact. So I like that view of the world, and I think that there's still mileage in the method. Okay, but still, that poses a question. Might structure be partly identifiable in the PSMC model? And actually, I posed this in the stats department in Oxford. I gave a lecture, but it's the same slide. I haven't changed it a few years ago. So the inferred values have dimension t, which is the number of time bins. Okay, but the transition matrix in the model has dimension t squared. We showed that. It's calculated. It's a t squared thing, but it's got just these t free parameters. And in fact, Kelly Harris's speed up, used this fact that it's only inherently got t free parameters. So we derive it from theory, but maybe for a structured population, we could be freer. Maybe there's something we could do with these extra parameters in the transition matrix. So maybe we could run the whole hidden Markov model, but free up our requirement that the transition values are derived just from one set of n's. And of course, we'll lose Kelly's nice speed up, but still, maybe we can actually infer structure. So Trevor started looking at this when he started as a PhD student, and he showed that, if you build a structured model and you have mass migration, and I can't remember, this is an old slide, exactly what happened here, but there was mass migration in some region, and we got transitions which looked very different from the ones you get with a panmictic model. Okay, so that looked hopeful. So then he went through a set of theory, and he added a pulse ghost migration into the model. So where you have a split time, there's a second population that splits, and then at some point we have a pulse back from it, and we never observe the bottom of B. So we have essentially two routes to get to our sampled population, either direct or into the ghost and back again. So these are the things you have to consider. You start at T. There's a recombination event at time U on that branch. It can either join back to the other branch at a lower point, or it can join back to the same branch or higher up to the parent. That's in panmixia. But in structure, you have to add the case that you get a coalescent event and it joins up into the other path, or that there's a set of alternative paths to consider. And there's a whole lot of maths, which I've made so small that nobody can even attempt to read it, which is a good thing. And it took Trevor some time, but we all believe it's correct now. So there's a lot. And one thing that makes us feel that it's correct is that things really do sit directly on simulated data. He went through all that. And so is this worth anything? That was the big question. So we created two models, one which was a structured model, and then we looked at what the implied implied NE was under the panmictic model for that structured model. And so we could make a panmictic model which had exactly the same NE to it, so they're matched. So it's from the sort of point of view of panmictic PSMC, these things are identical. There's no difference between them. But if we look at the two transition matrices, they're quite different, and not surprisingly, in ways that are not surprising. We have fewer transitions. We sort of block transitions in the structured phase, and we gain them in other phases and in the matrix. So there's certainly differences there. So can we do inference? So yes, you can identify structure. There's sort of one set of experiments on the left and one on the right, which I just didn't have the log likelihoods for this set of experiments, but there's a clear log likelihood difference between the two models, which is good, so they look separable. You can identify fairly well, so in solid are the simulated times, and in sort of paler colors are the inferred ones. So you can identify fairly well the split times, and one of the populations mirrors a change in population size, but the other one kind of dribbles off to not quite the right thing. So it's got weak power to identify all the parameters, even in if you allow everything to be free here. That's the essential message. Also, there's an identifiability problem about which is A and which is B, because there's two populations and they have to come back together again, but that's not really a worry, I don't think. So, in fact, the way that it behaves better, this model, and it's clearly just a sort of step, but is to tie one population as being to tie them so that there's only one free parameter in the relative size of the populations. Okay, but given that, we can apply it to human data, and Trevor did. And when we do that on this set of individuals, as listed here, so these are all HGDP, full genome sequenced samples. In each case, we get a clear best fit, which is structured, with structure starting with separation about 1 million years ago and finishing about 200,000 years ago. It's pretty consistent. The split fraction is about 25%. So it looks as though there was, there's support there for a model where this lump, which we see, wasn't due to a sudden increase in population size, which would have been, is at an interesting time in human history. It's at the time when, we're all told, that modern humans as a species arose, and all sorts of interesting cultural things started to change. So, in a way, if a new species is happening, you might think that there should be a bottleneck. That would be the first, if you're starting something new, that would be a natural assumption. But we saw this peak there, and the peak seems to be explained by structure, and the underlying population was not as large as that. Okay. And I think this is pretty closely related to Aaron Ragsdale's work and paper, and there's a poster in the next session, so actually I'm looking forward to talking to him about it. But I wanted to point that out. It was published last year. Okay, so that's interesting. But in a way, it's a sort of indirect it's a bit of a hack, really, because you're still using this panmictic model, but allowing the transition matrix to be free, not according to the model. So it's a sort of broken model. So the right thing to do is clearly to build the structure into the model. And Trevor has a new thing, SPSMC or something, structured paths. And so there's an additional set of states, essentially. There are multiple states, because there's either both paths can go through A, or both can go through B, or one can go through A and one through B. And you can just have all those as states in your model and work out the transitions between those states, and that's clearly the better thing to do. And that's underway, and I'm not going to show you more from that, but I think that's promising. Although it's still a limited set of, more complicated things no doubt happen. But if we allow ourselves to have everything structured in every way, it's complicated. Though I liked Nate Pope's talk yesterday, which possibly is a really nice way to get at these questions. Anyway, I have 3 minutes left. That's not good. Return to the human PSMC plot. Well, it had two bumps. There's this bump here, and you may have noticed there's another bump back here. And this is a particularly clean version of these plots. And early on, the first ones I showed you were sort of messy there, and people said, well, we can't really say anything. But actually, if you take a bit more care with modern data, you do seem to see another bump, and it's at about 5.5 million years. And that's kind of interesting, because that's the time of separation of humans and chimps, roughly. So you might say, how on earth can we really look back 5.5 million years? Well, if you look under these models, these are all fit models from different individuals, at how much of the genome is uncoalesced, how much is of course, when you're going along backwards in time, parts of the genome are coalescing all the time. That's what's giving us all this information. Is there really anything left? And that's why we can't go back beyond some point, because everything will have coalesced by then. But under these models, about 1% of the genome is uncoalesced at 5.5 million years, as you can see here. Maybe you can even stretch out to 10 million years. Who knows? It gets a bit messy. But 1% of the genome is quite a lot. It's a couple of hundred megabases. And it's, as a sanity check, with the effective population size for modern humans with the current mutation rate, with this mutation rate, is about 20,000. The mean time to coalescence is about 2NE, 40,000 times 30, 1.2 million years. E to the minus 5.5 over 1.2 is 1%, so it's what you expect. And the mean population TMRCA is 2.5 million years, so there's a lot of TMRCAs back behind the human-chimp separation. So where are these old coalescing segments? We're a bit nervous about them. How far can we really look into this space? That's the question. They're typically far from genes. That might not be surprising, background selection and so on. They're not in repeats. They're not in duplicated sequence. They do appear to be in different populations. at the same places, but that's what you'd expect because these are all populations that share things. So I don't want to promise you they're real, but I think that so far, kicking the tires, they've looked reasonable. Okay. So if we see that in humans, do we see it in chimps and bonobos? Well, the data that we have, or that Trevor's been using, isn't as good. We don't have such good references. There are all these plots people have made for chimps and gorillas. There may be something around this time. There's also issues about what the correct scalings are for mutation rates and population sizes. So better data is coming soon. I don't think we want to draw very much from that. Orangs and chimps possibly look like they go back further, which might be good. Possibly something even further back at gorilla. You could be hopeful, but there's hints, and I think more to be done. There are other things. You can mess with the model. You can have the mutation rate change in the past, and then you get changes in size, or you can have variation, of which there no doubt is, and background and selection. So I think there's more to be done to sort this out. But it's kind of interesting, and we haven't really dug into whether we can see structure and model that structure. But I wanted to give a few thoughts at the end about speciation, because I think this idea about hybridization occurring at speciation is something which now I've come across several times. So I'm now going to go into my red flashing period and talk about speciation as a little bit at the end. So this slide, actually I was showing this to Molly, and apologies, I don't know where I got it from, and I think there could be people in the audience from whom I got this, I think, who were working on ILS and estimates of population size from ILS. So it's an old slide. These are estimated population sizes in modern populations of great apes. And they're all, of the order of 10. But if you go back to the common ancestors, you can see these are estimates, yes, made in a slightly different way, but under a pretty understood model. And they all come out much, much bigger, order of magnitude bigger. And that suggests that the shared ancestor chromosome, the common ancestors were 5 million years, which, is quite a long, it starts to make these things overlap. And that's why we see ILS often in these things. And people had noticed, Celine and Molly had noticed that also there were big estimates for NE in these ancestral population sizes. So I think, maybe these are all hybridization type events. We should really we think of ourselves as separate. We know that there were Denisovans and Neanderthals around. We know that there's lots of evidence now of ghost populations and other hominoids in the last million years who are now extinct. There's evidence from lithics and tools and skull shapes of other forms. We should probably think of more like a braid. Possibly not a complete braid of species, but species that could come together, and possibly with speciation happening often at hybridization events where interesting things got put together. And I remember I was once actually in the American Museum of Natural History in New York, and there was a picture that looked a bit like this with all sorts of skulls and dotted lines, and they really, I think, honestly don't know how all these things were related to each other. And there's this great desire to say this is on the lineage to man, and maybe several of them are on the lineage to man. And I don't think that view has really become standard. But we've been noticing similar things in other species in the last decade. So this is a little coda for a few slides. I've been working on Lake Malawi cichlids. These are all extremely different sorts of fish. There are hundreds of species in Lake Malawi. It's a big lake, hundreds of kilometers long. They've all separated within the last million years, so it's a classic example of speciation. It's a radiation of species. It's one of the biggest radiations in invertebrates. We made a new reference genome. We've got 2,000 sequences from 250 species and populations. This is the diversity within species, about just over 0.1%, so comparable to human. But this is the divergence between species, and you can see they overlap. So they're much more similar to each other than we are to chimp. Okay. So if we run PSMC on these, we get an interesting pattern. All sorts of things happen. These are all different species. But we see a similar thing with a sort of bump, and then they're all the same behind it. And that bump is at about 500,000 years, which is, if you calibrate it with the mutation rate that we measured, which is the time of this radiation. It's when they had a common ancestor. And so again, we've got the same thing. And actually, we have direct evidence of hybridization at the origin of these fish radiations. This is a nice paper from Joanna Meyer and Olli Seehausen in Lake Victoria, where they found two lineages which are still extant and showed that they contributed to founding the Lake Victoria radiation. And Hannes Svardal with me did the same, and we found direct evidence of a hybridization from this lineage, which is 3.5 or over 3 million years diverged from this one, contributing about 20 to 25% of the gene content. And when we look within some people said, well, maybe that's what gave rise to the diversity, that some species got more hybridization than others, but that's not the case. I'm not going to go through this plot because I'm over time, but essentially these are all the different initial subgroups, clades, within Lake Malawi, and there's no differential, well, there's very minimal differential contribution between them. So the F statistics are 100-fold weaker between them than they are for the foundation. On the other hand, if we look at the variants which came from those two different lineages, they've been assorted on a variant-by-variant basis more than the other diversity in the system. So it looks as though there has been differential selection in the radiation compared to in hybridization-derived variants. And there's some controls in here, but this is this blue pot. So these are things that are in the proportion of FST in the global top 1%. Okay. So there's a message there that it looks as though, in this system and other systems, we can see increasing evidence that you can get hybridization events between things that are quite distinct, which you'd have thought would be quite incompatible, and maybe are incompatible, but which can contribute genetic material, which in some sense gives you a pick and mix possibility for new species to occur by taking different things. So the picture here is from Ole and Joanna's work, is that there's a hybrid swarm. There's a mix of alleles, and you can sort of recombine and sort them out and pick sets like an identikit system to make the fish of your dreams. And there's more that I can't talk about, so I'll conclude. So the conclusions are, we're on the cusp of having vast amounts of new high-quality genome data from across the tree of life. And in particular, we've got lots of diploid genomes of a huge variety of species. Given two sequences, PSMC is a really kind of natural population genetic model. It's a good approximation to the full coalescent with recombination, and we can also integrate over all the uncertainty in it, which is something which is a kind of, great opportunity, and so we can use that in multiple ways. I don't think we're at the end of using it. Despite being an old model, there's new ways to do it. I've shown you a couple to use it. And finally, that we're seeing more and more evidence for structure and hybridization associated with speciation. And that's it. And this is my group, punting, and last week we had a retreat in the cold of the East Anglian coast. Trevor is here, wasn't at the meeting. There's Regev, and Hannes has his own lab in Antwerp. Thank you.
Okay, we have time for a few questions. Someone has the mic there, and then there's a question over there.
So thanks, that was fantastic. I wanted to bring up the hybrid speciation stuff at the end. So there's sort of one way of looking at it that's a sort of bucolic thing, and you hybridize and it's wonderful, you get new nice things. But we've heard a lot of talks about kind of variants of fitness. So could it be the kind of opposite, that what happens is, you've been apart for a long time and then you hybridize, and that creates a massive drop in average in fitness because everyone's doing their own thing. And so it's kind of a Wallace effect thing where you drive to speciate to kind of reduce your variance again. So you want to isolate from all of this horror. of hybridization?
Yeah, so I guess there's two places. I think it can happen in different ways, and I should say my view about speciation is that there isn't one secret. I'm not saying that all speciation happens by hybridization. I think that there's just lots of different ways that things can separate, and you can only really tell after they've done so and they're properly separated that they have done. So one way is if there's kind of nothing there, you get some things in. Maybe there's only one of each. That's all they can do. And then, yes, as rapidly as possible, they speciate and fix the key, throw out the incompatible alleles. So that's, I think, the sort of thing you're saying. But also there are increasingly cases where in an established system, so there's some slides I took out, thank God, which say that one of the things that happened first in Malawi is that there was a deep, there were these open water fish, and there was a deep form that developed lineage. And then there were these ones that lived on the shore, and then there's strong evidence that there was hybridization between the deep open water fish and the shore fish, and that gave rise to a set of species which are benthic. They live on the bottom, but at deeper depths. They kind of acquired a set of alleles which are good for deep depth, and that opened up a new ecological niche which wasn't currently being used. And I think there's evidence, of kind of creating opportunity by stealing things. But maybe you only get a few things that way. It's a bit like the high altitude, in the EPAS-1 in Denisovans, coming from Denisovans into Tibetans. So, yeah.
Question back there.
Hi, Richard. Thanks for the nice talk. I was wondering about that. At 5.5 million years, the PSMC mutation model is not very accurate, I think, right? It's really more of a phylogenetic timescale. So I was wondering if you've considered how that could be biasing things, or did you actually use a different mutation model?
The mutation model?
Yeah, so you have to assume that 2t theta is like much less than 1, right? But I think if t is a couple 100,000 generations, then...
I think it's still, you don't have any, mu is,
I think
2t mu is, you're getting recurrent mutations at CPGs, no doubt. You get those even within the human coalescence. Well, this is within the human coalescence. But you're not saturating by a long, long way. So maybe, you could take more care over multiple. I don't think the multiple mutation thing is actually going to affect this particular model that much. It will matter for other things you would be interested in. But there's going to be a trade-off. We have thought of, we did look at just using transversions, but then, of course, you lose data that way, so there's trade-offs. So there's more to discuss, yes, in the model.
There's a question back there. I don't know. A couple questions back there, actually.
Can't see. But.
Thanks for the talk. The empirical observation of gamma-like behavior for pairwise coalescent times is fascinating. Do you have any commentary on a mathematical intuition or argumentation for that?
Well, it's the conjugate distribution. It's a very natural thing. Yeah, it's not a surprise. I'm not a good enough statistician. There are other people in the audience who can explain why it's the better thing. So I would embarrass myself to give you the mathematical detail, but it's the natural choice, actually. I'll say that, and it behaves well for those reasons.
Hi, thank you very much for a thrilling talk. I've tried to work with some of the programs that you have developed and talked about. But for some of us who are working with ancient DNA, the biggest problem is that our very low coverage genomes, that we cannot rely on genotypes. We have to rely on genotype likelihoods or imputation. And imputation becomes more difficult if you don't have humans. There was this, trying to use PSMC, there was this program, NGS PSMC, that was developed 6 years ago, but it's still in beta, doesn't allow bootstrapping, and not any publications is working with it. Do you envision or see any more advances of PSMC working with genotype likelihoods in the future? Are you interested in that, or do you know any?
So there's no reason not to work with genotype likelihoods except for speed. It would slow down all these fast things, but if you care about it, that absolutely that is okay. I'm strongly in favor of using genotype likelihoods rather than, of course, if you use pseudo-haploid sampling, then you wouldn't, you'd never get any differences. But you need a certain amount of, you need reasonable coverage. You can't do this off sub 1 or even 1x, sub 1x coverage. But in terms of implementation, I'm not the right person to ask, actually. No, we're not doing that at the moment. I'm almost going the other way of wanting to use, a very nice, really high quality new assembly data. It does remind me, though, I wanted to say, so there's a key parameter ratio, theta over rho, or the mutation to the recombination rate. And essentially you've got recombination events happening along the genome and mutation events, and you need enough mutation events to get observations. So you want that. In humans it's about 1, and you can work a little under 1. But unfortunately a lot of species that have small genomes and have low mutation rates, they have high recombination rates, and they can have at least 1 or maybe 2 orders of magnitude higher recombination to mutation rates. And I think in those cases, people tend to, I see people running PSMC, and I think it's meaningless. Actually, I think what you're seeing then is an effect of selection. You get wavy lines. And I think that that's a message I forgot to put in the talk that is important, that you have to think that you make sure that you actually have enough information in the data set for it to be meaningful.
Are there any other questions? Okay, well, if there are no other questions, first let's thank Richard again.